if I could just have everyone's attention for just a second. We're about to start our luncheon panel. Of course, please feel free to keep eating. We'll be able to talk over that. First of all, thanks so much for all being here today. We have a great group of panelists, and I think we're going to have a very, very interesting conversation. My name is Sai Mukherjee. I'm a healthcare reporter for Fortune magazine. I feel like I need to get this on the record. I am not Pulitzer Prize winning author and oncologist Sid Mukherjee. I've gotten that a couple times. I wish I were, but you know, that's life. Anyways, so luckily we do have an extremely distinguished group of panelists today. First, we have Dr. Laura Esserman of the University of California at San Francisco. She is the director of UCSF's Breast Care Center. We have Dr. Feng Zhang, who is of the Broad Institute and the McGovern Institute and is best known for his pioneering work in CRISPR gene editing. And we have Bhibash Mukhopadhyay of New Enterprise Associates. So, let's get rolling with this. Let me start with a very broad question for all three of you. And let's start with Laura and go down to Bhibash. When it comes to innovation in the cancer care space, in the last 10 years, what would you say has been the biggest advance, and what have we failed to achieve?
Well, it's a tough question. I would say, I think that right now some of the most important advances are in the area of immuno-oncology. And I think finally understanding how to stimulate the right cancers. I think perhaps, let me take that back. I think the most important thing that we have achieved, we have learned over the course of the last 2 decades to understand that cancer is not one disease. It is a spectrum of conditions that range from ultra low risk, that probably shouldn't even be called cancer, to those that are very aggressive. And I think we also now understand that there are cancers, cancers not only differ in their risk, but they differ in their timing of risk. And risk does not mean that everything should be thrown at them. More is not always better. The targeted is probably better, but you have to understand what you're treating. And I would say perhaps our biggest failure is not to design our trials and with that in mind to start with, and with the intention that we will take a risk and use agents earlier in the course of care, that the testing should be driven to the earlier phase of cancer treatment when cure is possible, and to really be innovative, to be willing to take the risk to do new things and drop some of the toxic therapies, and to really develop the early indicators of response like we have in cardiology with blood pressure or in AIDS. I think we are really at that moment. We can talk about that later. I think we are at a sea change, and new things really are possible. But we have to be intensely and fiercely data-driven, and we have to work collaboratively to make this sea change happen. And it's quite possible. I think we are at an inflection point, a transformation point in the industry.
I absolutely agree with everything that Laura said. I think in terms of treating cancer, we have made a tremendous amount of progress in the sciences in terms of understanding some of the mechanisms, the causes of cancer, and recognizing that cancer is not a single disease, it's many different types of diseases, and understanding the cause for each one of those types of cancer. I think we have made a lot of progress, but there's still a long way to go, because even for some of the most robust therapeutics right now, resistance can develop over time, and also in a lot of populations it will only be responsive in 40% or 60% of patients, and so there's still a large minority that is not responsive to those treatments. But with advances in technology, ways to be able to sequence the DNA, ways to be able to modify cells, that's beginning to allow us to start to hone into the mechanism, and that makes the therapeutics much more specific and targeted, so that we're not using chemotherapy and then killing many, many more cells than what we really just need to address in a specific patient. I think the area that still a lot needs to happen is in the area of early detection. For cancer, the earlier we can detect the presence of cancer, the better the outcome will be. And a lot of progress has been made, but really significantly more progress needs to be made on that front. And so I think that's one of the areas where a lot of effort really needs to be invested.
So let's make the answer to the first question unanimous, that I agree with the premise here that a lot has been known about the pathology and new mechanisms, new biology, new approaches stemming from understanding that pathology is coming online every day. And that's fabulous. If you go back to, dig up any journal paper from like mid-80s, let's say, that's beyond the time window that Sai suggested, but still, if you do that thought experiment, the depth and granularity of understanding of cancer that you see a snapshot of in those scientific papers versus what you see today is probably 100 to 1,000-fold more granular and much better. So that's an enormous scientific progress if you think about it. If you zoom out and last 15, 20 years, the space has undergone a sea of change. And that's excellent. That forms the basis of developing new drugs, developing new approaches. What hasn't happened as much, unfortunately, is that we still don't know. So sort of flip the card on the other side. On one hand, there is progress. On the other hand, there is still miles to go before you really understand what's going on at a molecular and pathologic level, and therefore can translate that into treatments. Hopefully that pace is catching up, and there are data points to argue for that it is indeed. Concurrent to that, what has also not happened, I think, is in some ways to extending Laura's point about patients still die. So unless that's fully under control and people don't have to die, I don't think any of our jobs in this room and beyond in the cancer care community is done.
So Laura, excuse me, Laura, I noticed that you were shaking your head a little bit during Feng's response. Do you have a reply to what he was saying?
Well, I think we want to be careful and not simplify the idea of early detection. Everyone likes to say that early detection saves lives, but quite frankly, it's more complicated than that. And part of the problem with screening is that it has really surfaced a reservoir of disease that is extremely low risk. And if you don't distinguish those cancers that are ultra low risk, you will overtreat these people, and the screening can cause more harm than good. And this has been, I think this is really this is actually very scientific and data driven. That, in prostate cancer, in fact, it has been so much so that screening has fallen from favor. But I think you don't want to throw the baby out with the bathwater, and screening really shouldn't be, cancer, yes or no. It should be cancer and what kind. We want to be looking for consequential cancers, not finding the inconsequential cancers. So I think so we actually are running a big trial called the Wisdom Study, Women Informed to Screen Depending on Measures of Risk. And we're trying to find out whether personalized screening versus annual screenings instead of age-based, that we really test on the basis of people's risk factors and assign them an age to start and an age to stop and a time, and a frequency of screening and a modality of screening. And the hope is that we will figure out a better way forward. We don't want to keep doing the same old thing we've done. That's no way to make an advance. And just looking for cancer may not be the right thing, but we can figure out who's at risk for what kind of cancer. In I-SPY, our trial where we're looking at the women at high risk for early stage, early recurrence breast cancers, most of those people are found between screens. Our screening isn't working for it. So there, yes, we may need to find better ways to find it, but we want to understand the biology better so that we understand what to do. More isn't better. More is just more. So we want to be very smart about targeting our understanding in screening and prevention and not just do more stuff and find more things. We have to really understand what kind of cancer you're finding. And that's another reason why I think we have to really do a better job of redefining cancer in 2018, 2020, using our scientific tools to say, if something is not likely to kill you ever, don't call it cancer. right? Makes people rush into procedures that they don't need and frightens them unnecessarily. But for those, and it doesn't help us cure the people who have the really serious cancers that will kill them. It just gums up the denominator.
So if I may follow up on that, completely agree with that sentiment. Ultimately boils down to what are you screening for and to what end, right? And to that end, in other diseases, we have this concept of a natural history of progression of disease. Has a similar effort been undertaken in a very systematic way across different types of cancer? And if not, why not?
Well, I think it's, so we actually been doing this. We actually are very lucky to work with groups in Sweden where they have long-term data. This is actually something, we can get into that too. This should be the way in which we practice medicine. We should not be allowed to practice without having long-term outcomes. But looking at that follow-up, you can actually set thresholds and set molecular markers that actually define an ultra-low risk group of cancers that probably a third of screen-detected cancers fit into. And by looking at the hallmarks of cancer and looking at those gene sets, we can start to understand, is this like normal, or is this, is this a new way to define what is cancer? And we're starting to look across prostate and thyroid cancers to try and understand, is this that same set of genes? I think this is, again, an important moment for transformation in the industry to say to come up and use these new tools to understand, what is cancer? And then really be very specific about the different types of cancer where you can not only intervene more specifically, but then eventually get to think about what's causing them and how to prevent it.
Okay, so Feng, next question for you. You are one of the pioneers of CRISPR, which is in the news all the time now. There's, I believe, a TV show being made about CRISPR. There was that Dwayne "The Rock" Johnson movie about animals being, mutated into giants with CRISPR. So it's pretty much hit the mainstream at this point, but I think it's safe to say that a couple of years ago that kind of widespread knowledge of this technology didn't exist. Can you discuss the process of innovation that actually led to the pioneering work that you've done on this, as have others such as, obviously, your rivals over at UC Berkeley and whatnot?
And remember, I'm from a UC.
So one of the really exciting advances in biology in the past decade or so has been the completion of the human genome. So our cells have something called a genome, and that's composed of DNA. And that DNA has 4 letters and is 3 billion letters long, and that's our genome. So our ability to sequence DNA has really exponentially increased. So now the cost for sequencing a whole genome is about $1,000 and even less. And so from that, that means we can start to sequence many genomes and start to compare them to see what are the differences across these different genomes. And some of those differences contribute to disease, and others may increase health by reducing disease risk. So then one of the really tantalizing ideas is can we go into our DNA, know what sequence we have, and if it's deleterious, fix it, and if we can also put in potentially beneficial genetic differences, then we can further increase health. So that's kind of the tantalizing idea. So that idea has been worked on by many, many researchers over several decades now. And so the main idea behind that stems from a concept of using what's called DNA nucleases, which are these proteins that can go and chop up DNA. And if you can program it to chop up DNA only at the place that you want to fix it, then you can precisely change it. So our ability to then program this nuclease to chop DNA is something that has gradually increased. There are several other technologies that came before CRISPR that includes something called zinc finger proteins or tail proteins. And the evolution of this technology really has to do with how easy and how precisely can we reprogram this protein to recognize a new sequence. So the reason that CRISPR is really exciting is because it's one of the most easy to reprogram systems. All you have to do is know what sequence you want to recognize in the genome, what's the string of letters you want to search for, give it a new piece of RNA, and that RNA is simply the complement or is written to be opposite of the sequence of DNA that you're trying to find in the genome. And by only giving that piece of RNA, then you can make it recognize a specific gene in the genome and chop it. This system existed in the microbial world. It's a system that bacterial cells or archaeal cells use to defend themselves against viruses. And nature, in a way, it evolved and invented this way of using this protein to recognize new DNA sequences by very easily changing that RNA molecule. So by repurposing this system, then we were able to then develop a way to chop up human DNA. If you harness the bacterial protein, engineer it, and optimize it so that it can work in a human cell, you can program it to recognize a mutation involved in a rare disease like sickle cell or cystic fibrosis. Or you can use it to modify immune cells to get it to recognize cancer cells better to develop immunotherapy. And so that's where this technology has really sort of evolved and emerged from a whole series of studies that led to realizing what we need to have. But I think there's a lot of talk about using CRISPR for treating disease. But I think one of the really exciting applications, in addition to treating disease, is to use it to understand disease more. CRISPR systems are so easy to program. You can use it to target every single gene in the genome all at once. There are 20,000 genes or so. You can design this to perturb one gene at a time, but all 20,000 genes within the same experiment. And by doing that, we can start to ask questions like, what are the genes that's involved in causing a cell to metastasize? What are the genes that cause a tumor cell to develop resistance against a specific drug? Or what are the genes that would make a cell more susceptible to a drug? And by doing that, we can start to figure out where are the pinpoints in these cancer cells? Where are the levers that we can pull to make it react more or react less? And based on that understanding, develop better therapeutics. So those are just some of the things, but I think it's one of those tools that we can combine with a whole cadre of new technologies that are being developed in biotechnology to improve patient care.
All right, Bibhash, next question is for you. As I understand it, you have some insight into, on the venture capital side of things, where the money flows when it comes to cancer research has changed. Why don't you tell us a little bit about that evolution?
Sure. So one framework to think about how you build companies, which in turn builds drugs, is that it requires the confluence of three things that have to happen, one of which missing wouldn't make the endeavor successful. These are science, people, and so scientific capital, human capital, and of course, financial capital. So as venture firms, we try to identify the convergence of these three and try to build companies which have the right vision and the execution capability to do drug development. As you heard and got a snapshot from Fang, the idea of scientific progress has been tremendous over the last 20 years or so. The people who know and understand drug discovery and development, have learned their chops. We have a term, we call it the scars of drug development. Unless you have done it, unless you have failed at doing it, you do not really become a drug developer. Sort of take the next step from identifying a target to getting to a drug. It's a pretty arduous, long, very expensive process. And some people can do it very well. So to top that, ultimately, the convergence of these three become very important. And if you just take a snapshot of what has been going on in the industry for the last, let's say, 20 years, when you had, taxane and a couple of other DNA damaging agents or DNA repair agents as the primary modality of cancer treatments in the mid-80s. Fast forward that to now, when you have extremely sophisticated immunotherapy, as Laura alluded to in her first statement of cancer, in which you engage the body's own immune system, turn it upon the cancer which is infesting you, and try to remedy and cure some of it. So in this process, multiple drugs have come, multiple approaches have come, where it used to be one single small molecule IV delivered agent. Now it has, you can arm a virus to attack your cancer. You can arm your own, you can have different antibodies targeted against very specific cell subpopulations. You can take out cells from your own body, modify it, put it back in, and help attack the cancer. So all these steps that have happened over the years required a very deliberate and detailed drug development process, which has been going on. So just take a snapshot over the last several years. For example, in 2017, I think there were 46 drugs approved by the FDA, a record. And out of those, either 16 or 18, I can't recall exactly, were oncology drugs. Isn't that amazing? 18 oncology drugs approved in 1 year, which were probably in development for the last 20 years. If you take a step back, that's momentous progress. Hasn't happened ever before in the industry. Yet it's possible now. The number of drugs in development that are in the pipeline, they go through the different stages of human clinical trials, is also exponentially increased. And so, all of this is also enabled by the fact that over the last several, 7, 8 years or so, we have had an incredible influx of capital that enabled all of this to happen. Around 180 companies went public in the last, 4 or 5 years in that window. Show me another time point in history where that has happened. $2.8 billion, 2.6 actually, billion dollars have flown into oncology-focused companies. So these numbers, is part of me quoting these numbers is to shock and awe you that enormous amount of capital is going in, enormous scientific discoveries are being done, and the drugs that are now in development will, a subset of them at least, will come to fruition 5, 6, 7 years down the line. And this is momentous time in history to sort of step back and think through that. And all of this is possible by the convergence of, the three things that I mentioned. So I personally think that as investor, as a scientist turned investor, there probably has not been a better time point in the history of drug development to be sitting on. And the future is enormously promising. Now, does that mean that everything is perfect? No. Lots of drugs fail in the phase 3 trials. Lots of hiccups happen along the road. But that's a part of the process. That's the fun of drug discovery.
Laura, you look like you had something to say about that as well.
I do. I think actually to realize the potential of all this, we need really a very different approach to clinical trials. And I think this is now is the time. And I can use the experience of running the I-SPY trial program over the last 20 years as an example. I think, as I said earlier, that the backbone of the I-SPY trial was to think again to say, first, that we want to try and find the right drugs for the right patients at the right time. And the backbone of it is to say, first, to try and test the drugs where they matter most. Gleevec, works in the accelerated phase, not in blast crisis. I would say that we put so much of our drug development into the metastatic phase, but in fact, when you invest in earlier stages of disease, you can actually cure people. So the trick is, again, not to treat everyone, but to really focus on a group of people. So what we did was we said, we're going to use breast cancer as an example. And we had a very long scientific experiment where we said we were going to focus on a group of people who were in the early stage, high risk for early recurrence, using biomarkers to identify those patients. And in phase 1, we all worked together to establish ways of common clinical and pathology and imaging processes, to standardize those processes, to develop a culture of collaboration, data sharing, and innovation. The second phase, we added an adaptive platform trial to be able to take one trial where you bring many drugs in, and not every time you need, when you think about all the time and money that we waste in trials, every time you run a phase 2 trial, you start again. and you give up all your infrastructure and start all over. Same thing, 2.5, 3 years sometimes to get drugs. That is a total waste of time. It's the same process. So you start again, you start and have one common platform. You find different ways to engage investigators and scientists so they get credit in different ways, but all working together in a team-like way. You have pre-competitive collaboration where industry can come together and share information at the critical time. And at the time, and the idea is you change the order of therapy so that you can learn early in the course of disease whether or not it's worked. This business of, I'm a surgeon, and I can say when someone is at risk to die of their disease, my operating on them is not going to save their life. You have to recognize what you contribute, what you don't, and get out of the way. What I can do instead is say, wait, if we give you the systemic treatment up front, we can see whether this treatment will work and whether your tumor will shrink. For the fast-growing tumors, that works. And of course, you'd like to get every person to have the point where their tumor goes away before they go to the operating room, but we didn't have enough drugs at the time. And so we have now put in 15 drugs. We will be 19 by the end of the year, but we have really surfaced a number of drugs by class that really change, double and triple the chance of getting to a complete response. And perhaps the most important thing that we've done, we've stuck to it, and we've got the data to show that if you get a complete response in this particular group of patients, that your chance of having distant disease-free survival is 95%. And that is true across every subtype within that high-risk group. It's true whether it's a new therapeutic, people thought maybe the immunotherapies wouldn't work, or it doesn't matter if it's a standard therapy or not. This is really critical because it's a sea change that propels us into our next phase, which is now to say, we've been running the trial, we've created this learning system with Bayesian adaptive design and platform design, and Anna Barker really helped us architect a way to do this with Janet Woodcock, initially the FNIH, and now Quantum Leap Healthcare Collaborative as our sponsor. What we are really able to do, will be able to do is say we can get every patient to a complete response. This is important. If you can get to a complete response, do you need to use drugs like adriamycin? Do you need to give people toxic therapies? All those great drugs in the pipeline, they need to see their day in court. So once you have one drug that works, is anyone going to be willing to try innovative things? You have to have a structure that helps you do that. And you have to be able to have a way to use the science to help you learn. So what we're going to do is say, now we're going to be able to adapt and give people multiple shots on goal. We're so excited to be working with the FDA on this, Janet Woodcock and Rick Pazder and Tatiana Prowl, to be able to say, look, we'll bring in the new drugs that are more targeted and less toxic. If they work, great. And you get a complete response, you go to the OR, and you're done. If not, you can be assigned the next drug, the best drug for your particular subtype that worked in I-SPY 2. If it works, you go to the OR. If that doesn't work, you then will try new things. And maybe that's where you're learning from your CRISPR, or you're learning from the targeted therapies where you're targeting emergence of resistance. We need learning systems and frameworks to help us learn in much faster ways. And this process is what's going to help us get every patient to the right response. To be able to say to someone, look, this is, instead of testing drug A versus drug B, and I think someone described what the randomized trial process was, and you can't always do that because you don't know what's going to work, but if you know that something is 2 or 3 times better, why are we going to give someone the control? And the idea that we actually have the controls, we've been running this experiment for 8 years, so we'll be able to evolve and say we can still use that information. This process is scalable and replicable. There's a lot of science in the management of trials that needs to be turned into an engine for learning and discovery. And quite frankly, our trials need to look more like care, and our care needs to look more like trials. And this is the future that we should all be getting to, where we're collecting the mission-critical data at the point of care, finding out if they can get on these trials to get every patient to this best endpoint. And I'm telling you that if we actually have the imagination, the effort, and are willing to take the risk, we can get everyone. In 5 years, we can achieve 90% of patients with high-risk early-stage breast cancer to a complete response in 5 years without standard chemotherapy. I'm sure we can do it. But you have to also be willing to have a culture of innovation where you're saying, I'm willing to take a chance to do something different because what we are doing today is not going to get us there. You know, when I look around, if people are still dying of breast cancer. Okay, we're still making people sick and giving them toxic therapies. If we want a different world, we have to be willing to do something different. And I know our patients are willing to do it. I know our group at iSpy is willing to do it. I think this is a kind of model that we should all really try and scale and replicate.
All right, great. We are unfortunately... So we're running a little bit short on time. I'm sure we could continue this discussion for like another 2 hours. But we did want to give some of the audience members here a chance to ask questions if they had any. We don't have too much time, but if anyone does have a question, please just raise your hand and we'll get the mic over to you. Probably just one or two, yeah.
A question for Dr. Esserman. Given the potential of adaptive trials like iSpy, why isn't industry embracing them more?
They're actually hard to run. They're expensive. Actually, you do have to do a lot of work up front. It requires a lot of collaboration and breaking down of silos that we don't otherwise break down. You know, I hope this will be a sea change and people can see now that we have the data that demonstrates that they can work, but it's going to require a lot of work. Change is hard. That's all I can say.
I would like to add one point that builds on Bibash's point about investment. So there's an enormous amount of funding that has gone into the later stages of therapeutic development, but not enough funding is going into the early stages that fuel the pipeline for this innovation. So investment in basic science research, and these are the research that led to the development of innovative technologies like sequencing or gene editing or all of these things. And that early stage investment is really not enough. Everybody is focused on making money by investing in the later stage translational work, but so much early work is not being invested in. And the second thing is science education. I'm a member of the board of trustees for an organization called the Society for Science and Public, and it organizes a phenomenal program called the International Science and Engineering Fair. For the past 20 years or so, Intel Corporation has been the major title sponsor of this program, which has benefited kids who are interested in science and encouraged learning of science for more than 2 decades. Intel is ending their title sponsorship, and we have been having an incredibly difficult time finding any corporation to sponsor this program. It's a phenomenal program that has benefited, I think, even some of the members of the audience here. But the fact that we couldn't find a sponsor to sponsor this important program is a signal that we're not doing something right. Something is wrong about the way that we're focusing our investment on society. We need to nurture the next generation of scientists who are going to lead the development of new treatments, who are going to lead the translation of these new discoveries. And the lack of that really is troubling as well. So I just wanted to add those 2 points.
Okay, I've been given the flashing red light, which means that we're out of time. Let's give a round of applause to our panelists. All right, thank you so much for sticking with us, and I hope you enjoy the rest of the summit.